Okay, so thanks everyone for joining us today for another webinar by the International Weed Genomics Consortium. Today it's my pleasure to have Dr. Dana MacGregor with us leading the discussion. Dana is a weed molecular biologist with Rothamsted Research, and I will quickly read her bio here. Dana research covers a range of plant science disciplines, including plant physiology, weed biology, genomics, and gene function studies. Her focus is on uncovering the molecular mechanisms driving plant plasticity and resilience. Working with both model and weed species, Dana explores how plants perceive and adapt to a wide range of natural and human-induced stresses. She has contributed to our understanding of how plants interpret environmental cues, such as changes in light, temperature, and water availability to appropriately tune seed dormancy, root development, and circadian rhythms. Most recently, Dana's work explores the genetic and phenotypic diversity of kiwi species, including blackgrass, ryegrass, limbs quarters, and amaranth species. By quantifying weeds' plasticity and ability to resist weed controls, she defines which weedy traits make weeds hard to control in agroecosystems and better able to withstand environmental changes, thereby generating knowledge that will directly contribute to sustainable weed control and generate novel avenues for future-proofing crops and to climate change. So Dana, thanks so much for joining us today. It's a great pleasure. Good to see you. Just one reminder to the audience is that the recording will be available on the IWGC website, and the previous webinars are also available there. And a final reminder is that we have a Q&A tool here on Zoom, so please feel free to enter your questions, and then I'll read to Dana later at the end of the seminar. And as we discuss the questions, you can keep adding your questions there as well, and we will answer them as much as we can. Dana, thanks so much. Looking forward to your webinar today.
Thanks very much. I will try not to cover all of the things that Kaya just introduced, because that would be a very long seminar. But today what I thought I'd focus on is just quantifying and getting to the roots of waterlogging tolerance in blackgrass versus wheat. So this is a paper that we published relatively recently, and I will put the acknowledgements, and all of the links will come up through the slides. So hopefully that will help you to find more information if I haven't given you quite enough. I will do my very best to leave time at the end for questions, and I'm very happy to work through those. I am using two screens, so sorry if I'm looking a little bit in strange places. So just to start off with, in Kayo's introduction, there was lots and lots of mentions of the words weeds. And for me, weeds are just plants that humans don't like. They come in lots of different flavors and colors and situations, but essentially, for me, all of weeds just covers anything that humans don't like. And one thing, although we can all disagree as to whether or not some of these are weeds or not, one thing that we can all agree on is actually that weeds in the agroecosystem are particularly disliked. So weeds that are growing in and amongst our crop fields are ones where pretty much everybody agrees shouldn't be there. Although this is a very beautiful picture, and it shows you what happens when you don't apply any herbicides in an agricultural setting. It isn't something that's a particularly useful place for us to make crops so that we can have enough food to sustain our futures. Now, although it's interesting that we can all agree that weeds are disliked, one thing that is interesting is that just because we don't like something doesn't mean that we should have an entire research program about it. So I wanted to start off by asking the very first question of why should we be researching agricultural weeds? And the way in which I justify my research program is twofold. One, there's a clear correlation between decreasing crop yield and increase in weed abundance. I'm showing you some data here from Richard that just shows that as you increase the amount of weeds that are in a field, your crop yield is going to decrease in a linear way. So this is one side of it. And on the other side of it, weeds are remarkably resilient plants. These are plants that can grow in a variety of different situations. They're really resilient to agricultural change, to climate change, to heat, drought, stress, all sorts of things. But they're also growing often, not like this plant in a parking lot, but also they're growing in our agricultural settings. So they're able to withstand the same pressures that our crop plants are growing. So this is sort of a two-faced side of things, where on one hand, in order to ensure food security, weeds must be controlled. We need to make sure that we have enough food from our fields and that we're up here on the yield ends, not down here on the yield ends. Now, the other side of things is that weeds are actually remarkably successful plants that grow under challenging conditions. I'm very much a plant person by nature and by training, and so therefore I can see that there are some huge benefits to understanding the traits that are allowing plants to resist these challenges so that we can figure out how this is working. In short, if we want to end hunger, achieve food security, and improve nutrition and promoting a sustainable agricultural within the agricultural field space that we have, removing weeds and understanding how weeds are surviving in this situation is a very good way to do this. So I just wanted to then focus on the different challenges that weeds are surviving in these sections. There's not very many things that can survive humans. Humans are usually pretty good at killing things. And if we think about it, weeds have survived our ancestral weed control challenges and our most modern weed control challenges. I see this as a very good thing because actually it helps us to figure out how weeds are going to survive in the agri-ecosystem in the future so that we can figure out how to control them and what they're doing to circumvent those challenges now. On the other side of it, they are growing in our agricultural fields, and these are the fields that are going to be facing tomorrow's climate change issues. So if we want to future-proof our crops by understanding how weeds are resisting the droughts, the waterlogging events, the heat, the cold, whatever it is that they're facing today, we can then learn from them to go towards tomorrow. So just to summarize this, weeds survive abiotic stresses, biotic stresses, chemical controls, and cultural controls. Today, just for the sake of time, I'm just going to focus on abiotic stresses and chemical controls. Now, in the UK, blackgrass, Alopeperis microsides, is our worst weed. I already showed you this picture here on the left, which is our very beautiful no herbicide situation. And here's a new picture on the right, which is another beautiful picture of a very clean monoculture of wheat growing actually not very far away from where I took that original photo. Now, what we should see is this picture here, if herbicides are working how they ought to be working. However, over-reliance on a limited number of herbicides and use of herbicides for many, many years on the same sorts of crops has led to the situation here in the middle, where what you see is there is actually wheat underneath all of this, and these heads sticking out above are blackgrass heads. Now, they're called blackgrass because the flowers and the heads have this sort of purple hue to them. So when you look out across the field, there's sort of a black haze that's floating above the crop, and hence blackgrass. Now, blackgrass is something that's a particularly bad weed in the UK. And in 2016, herbicide-resistant blackgrass was confirmed in over 20,000 arable farms across 37 different counties in the UK. These are the ones marked here in purple. And you can see that the purple is actually outnumbering the green in a massive way. Most of these counties are also where we're growing our wheat. So pretty much everywhere where there is wheat, there is herbicide-resistant blackgrass in the UK. It's not just a UK problem then, it is also across much of Western Europe. But today I'm just going to focus on what's happening here in the UK. And the data that I showed you originally of this correlation between crop yield and increased herbicide-resistant blackgrass heads was from blackgrass. So there's a clear correlation, a linear decrease in the amount of food that you can get from a field if blackgrass is there. Now, I don't particularly think in meters squared or in hectares per yield. What I think about is bread. So for me, just to put this into context, a 1 hectare field without blackgrass produces over 11,500 loaves of bread, while one with heavy blackgrass pressure only produces 4,600 loaves of bread. So again, reducing weed pressures is a good way to food security. Putting this into money, we're talking about 0.4 billion pounds of lost income for farmers due to just the one weed. So we can break that out into various costs per hectare across the country, but really what we're talking about is 1.2 billion loaves of bread per year. That's quite a significant contribution. And so hopefully what you can see is that it is very clearly an issue for us in terms of securing food security by removing blackgrass. But on the other hand, blackgrass is this plant that I showed you that's growing in the parking lot. It's growing wild all over the UK. Mostly you can only find it in agricultural settings, but it's also a riparian weed. It is naturalized in the UK, and we can talk about more of that during the discussion section. But just to focus on it in the agricultural setting, it survives abiotic stresses. It has non-target site herbicide resistance. It has target site herbicide resistance, and it can survive biotic stresses. This is a very resilient plant, and it's great for me in the lab because it's very difficult to kill. But why is it difficult to kill? What I'm trying to figure out is how is it able to survive all of these challenges? How is it able to survive the natural stresses that we're imposing upon it, as well as the human imposed stresses of things like herbicide application on a regular basis? Now, today, I'm really, again, just going to focus on the abiotic stress and a little bit about the non-target site resistances. There's a number of times where I'll pop up this QR code along the way, and that's just to link you out to additional resources if you want to learn more about the research that we are doing. And whenever I say we, what I mean is all of these people. So I have a huge number of published co-authors that I've worked with throughout the years. This is in my life as an Arabidopsis geneticist, also in my life as a weed biologist. And there's a number of people who I'm working with currently, and their names will appear as unpublished research that's going on in the background. So again, this is the QR code if you want to know more. And thanks to all of my funding and support through Growing Health and Rothamsted Research through this. I should also say that if you ever see this little PubMed, it's something that I've taken directly out of the literature, and I have nothing to do with it. Okay. So coming back to this idea that plants are surviving a number of challenges in the agri-ecosystem. One of the challenges that we know very well here in England is waterlogging. So this is a picture of a field that's very close to my house. And what you see here is a giant wet patch. Now, under that giant wet patch, there should be little wheat seedlings. Now, we all know that when wheat is waterlogged, what ends up happening is it goes yellow, it's really chlorotic, it's really unhealthy, and waterlogging and wheat do not mix very well. So the negative effects of waterlogging on wheat are very well established. To put some numbers on that, there's some evidence that demonstrates that yield loss due to waterlogging and wheat is somewhere between the range of 20 to 80%. So that's a massive yield reduction, and specific numbers on that. Here's a variety of different variables, including seed number spikes, seed weights, yields, spike numbers, etc. And as you increase the amount of days of waterlogging, each of these numbers decreases massively until a point where you end up with just 6 rather than 32. So these are massive changes, a massive decrease in the yield of wheat when waterlogging is happening. Now, going back to that same picture, what is underneath of that, if you go back a few weeks later, what you see is poor establishment of the wheat and a very fantastically happy blackgrass plant just sitting in there. Years, a couple of months later, again, what you see is this massive patch of blackgrass. Again, blackgrass is these longer, thinner heads poking out above. Now, it wasn't just us that was noticing that. Collaborators of mine here at Rothamsted noticed that these patches were happening across their surveys that they were doing. And when you measure the abundance of the blackgrass seedlings, what you see is a positive correlation in the fields that's associating with specific properties. So this was a paper that was published not too long ago. And what they did is they went out and they measured various fields across England. And they measured not only the abundance of blackgrass seedlings that were happening, but also various epiphytic drivers that were underpinning this. So things like soil moisture content, available phosphorus, clay content, pH. And essentially what they showed is that there's a clear correlation, there's a relationship between weed abundance and distribution of different soil properties. In short, the soil has a very high probability for waterlogging. So there's clear evidence that there's a correlation between where the weeds are and what the soil is. And we knew from the literature that wheat does not tolerate waterlogging. So all of this led us to ask whether or not the differences in waterlogging tolerance are driving this patchiness. So it was correlative, but is it causative? So we know that from the literature that wheat is intolerant. Is blackgrass tolerant? And is this tolerance then allowing blackgrass to tolerate the waterlogging better so that it can survive in these patches? Now, to start to work this out, we not only knew that these correlations were loss of yield, and we saw this sort of clear survivability within the blackgrass within these wet patches, but other sister species of blackgrass, which are things like Alloparis aruncius and Alloparis aqualis, are actually riparian species. I mentioned in the beginning that blackgrass can be found near to rivers and wild across the UK. These other species are definitely riparian species. So this all led us to hypothesize that blackgrass is going to be more tolerant to waterlogging than is wheat, but we needed to test that. In order to test that, what we did is we brought blackgrass into the lab. We made this wonderful setup, which took quite a lot of time to work out, to figure out whether or not we could systematically test the tolerances between species and within species for blackgrass. So what we see is that blackgrass itself, it's this tiny little weed. If we grow it in pots with a coating of sand and no bag, this is our control setting. Whereas if we just stick a plastic bag and quite a lot of duct tape underneath the bag, what we see is that we can totally flood these systems. These allow us to have this clear, nice ratio of control over control and a ratio of waterlogging over control. Now I'm saying a ratio because wheat is a lot bigger than blackgrass. And so in order to present these data in a way that seems really easy to understand, I'm going to be presenting ratios for each of these data sets. So the data looks like this. You have wheat on the left and blackgrass on the right. And when we're looking at the relative fresh weight, what you can see is that control is always at 1. That's by definition because it's a ratio. And for wheat, all of the values are less than 1. This means that the waterlogged wheat is smaller than its control counterpart. On the other hand, all of the blackgrass that we tested are all bigger. They're much, much bigger, even about 3-fold bigger than the ratio of what they were when they were not waterlogged. So this means that not only is blackgrass surviving the waterlogging, it is doing way better. This is allowing it to outcompete, giving it a competitive edge. Now, these are in early seedling development, which means that that difference is going to be manifest over the growing time. And actually, the amount of seeds that these guys are going to drop is way more than they would than the ones that were just on the well-buttered condition. So this is a very clear difference in fresh weight. It also manifested in things like height, where we can again see this clear difference between the wheat on the left and the blackgrass on the right. All the blackgrass are doing as well, if not better. At height, the blackgrass is really going to be tillering more than it is sending up one big tall tiller. So these differences are manifesting outward. The great thing about measuring height, though, is that it allowed us to completely recapitulate the tolerances and intolerances that were previously published in wheat. So here we have a setting, an experimental setup that allows us to recapitulate what we've seen in wheat, where we have some intolerant varieties and some tolerant varieties. And it allows us to test blackgrass as to why now it is doing what it's doing. So just to summarize this part really quickly, we know that wheat does not tolerate waterlogging and that it manifests in yield differences between a loss of 20 to 80%. And what we've been able to show is that, yes, blackgrass is more tolerant to waterlogging. This happens out in the field, and we can see it in the lab where there's a very clear correlation between whether or not you are blackgrass, you will survive, whereas if you're a wheat, you will not. So again, why is this happening? Just measuring that it is happening isn't quite enough. So what we did is I focused on just a few of these lines. We focused on Frument and Jackson, which are both winter wheats, and 2 populations of blackgrass, one of which that had been gathered from a field collected population, and one that's sort of a lab strain favorite of ours. This is one that is well known in the literature, and it's kind of just a great one to work with. So Peldon had a much higher biomass when we measured it. The Lola 103 was pretty much even, depending on whether it was waterlogged or not. Jackson is the tolerant, and Frument is the intolerant. So we then systematically worked through physiological traits, transcriptomic traits, and metabolomic traits. And I'm going to talk you through those for the rest of the talk today. So starting off with physiology, when we're talking about waterlogging, it's really where the water level is just to the junction between the top of the soil, so at the root shoot junction. So what happens when you're fully waterlogged is that it induces hypoxia stress in the roots. And so these roots do not have access to oxygen, and they start to die off because they can no longer do any of the normal cell division and growth that they want to do. Now in wheat, and we know from wheat and other plants that when they are waterlogged, what ends up happening is there's sort of 2 stress responses that happen. if you're totally flooded, so this is a massive flood where the water level is way above the level of the plant, so it would be up right here. There's a very systematic response that happens. That's not what I'm talking about at this point. What we're talking about is just when the water is at the root shoot junction, so just totally wet but not submerged. What we know about in this situation is the waterlogging. When the water level is just here, there's a formation of parenchyma, and there's very systematic production of things that lead to barriers of radial oxygen loss. What I mean by that is that the parenchyma actually are these open tube-like structures that go all the way down the roots. They open all the way out, allowing for gas exchange between the root, the top and the bottom of the root, so that the oxygen exchange can happen. So instead of a well-drained condition, these parenchyma would stop lower down into the root, not quite down to the root tip, and they're also quite a lot smaller, so the oxygen exchange is happening all the way along. Whereas under the waterlogged soil conditions, there's an opening all the way along. You can see it's a much more open root structure as compared to down here. So that's what happens in other systems. And essentially, if you think about that as a cartoon version, it's a little drinking straw that allows the bottom of the root to breathe the air above the water line and to have that gas exchange so that there's sort of an easy about to breathing. So we brought these plants into the lab, and when you look at wheat, we were able to recapitulate what we saw from the literature. So you go from a very nice, regularly ordered closed root structure, and under the waterlogging condition, you end up with these lovely little drinking straws that allow the gas exchange to move the air all the way down through the root to the tip. Blackgrass, on the other hand, had these drinking straws constitutively. So under all conditions, the blackgrass roots that we looked at had these big open pore structures under the control and under the waterlogging condition. Putting some number on that, we went from Jackson that had no parenchyma to lots and lots of parenchyma when waterlogged, whereas Peldon had parenchyma at all times and then had more parenchyma at all times. It wasn't just Peldon. Each of the blackgrass lines that we looked at had these constitutive parenchyma under the control conditions, and there were some changes in response to the waterlogging, but again, there were parenchyma all the way through. So in terms of physiology, when you have this waterlogging stress, there are clear differences in the heights of the above ground tissue. So this is manifesting that the plants are doing better. When you're a blackgrass, you're doing better when you're waterlogged as compared to wheat, and you're doing worse. And we've seen that there are clear differences in the root structures that are happening, whereas the wheat has to respond to the waterlogging and create these parenchyma, these drinking straw structures. Blackgrass already has them in place. So it can kind of avoid that hypoxia stress because it's already having the air exchange, the gas exchange from the top to the bottom of the root all the way through. So this is just sort of to summarize the physiology part of this. And so now we'll move on to the transcriptomics. At this point, I wanted to point out that actually our transcriptomics and metabolomic analyses were done 21 days after these guys had been flooded. So they'd been wrapped up and flooded completely for 21 days. So this is not an immediate response. This is not a knee-jerk response, but this is a real change in the physiology, a change in the transcriptomic profile, so that we can see how they had adapted to it, not how they were responding to it. So starting off with metabolomics, what we see is that we measured a variety of different metabolites that are sort of a standard profile of things to look at in response to stress. Now I'm showing you here Frument and Jackson, the wheat in the yellow and in the red and the yellow, and the blackgrass are in the green and the purple. And what you can see is that blackgrass kind of has a much smaller metabolic response than does the wheat. So when wheat is way up here at 5 and 6, blackgrass is really kicking around down here around 1 and 2. So just to zoom in on a few things, we knew from the literature that sugars ought to be up in wheat. They are. But what was really interesting was that we had some metabolites that were only specific to blackgrass. So looking at this more carefully, what we see is that blackgrass itself, again, as I said, there's a very much smaller metabolic response. And we have a few number of things that are actually statistically different in response to it. Many of these are actually downregulated in blackgrass response to waterlogging. There's a few that are up, and the 2 that are highlighted here in purple are the ones that are only in blackgrass. And you can see that actually they're down by half, or they're up by 1.4. So these are not massive metabolic responses that would be totally overcoming this. As I said, in the literature, we expected to see that sugars and some specific amino acids were up in wheat. These are. So for instance, fructose should have been up in Jackson from a paper published in 1995. And yes, we do see that Jackson has an increased level of fructose in compared to the waterlogging. Now, there's some really interesting things that have fallen out of this that we didn't expect, is that there's a few things that are different in Jackson compared to Fruman. Now, these might be playing a protective role because they're actually higher in the tolerant one compared to the intolerant one. So they might be a sort of response, a protective response compared to one of these. So we are still learning new things about wheat while we're still picking apart what is happening in blackgrass. So this, don't want to go too much more into this, into the details of this, but essentially, just to summarize, there's a metabolic response in wheat that is reliant on things like fructose, sucrose, raffinose, the sugars and the amino acids that are highlighted. And there's a small number of responsive elements in blackgrass that are different, but not really massive changes in what's happening. A similar thing was sort of seen with the transcriptomics. So we didn't have that many genes that were differentially expressed under waterlogging. So we had about 150 genes that were differentially expressed in wheat, but only about 50 genes that were differentially expressed in blackgrass. So this is interesting, but just to remind you, this is probably because this is not an immediate response, but something that's a constitutive response. So they have adapted to this waterlogging rather than just the sort of survival responses that are happening. The cool thing is when we look just at what's happening in the differentially expressed genes in wheat, many of these are leading to go terms. I'm sort of not expecting you to read these. Sorry about that. Many of these are things that we were expecting to see, including, for instance, the fourth from the top, which is a cellular response to hypoxia. So the genes that we're expecting to see differentially expressed in wheat are happening, and they are differentially expressed. And just to zoom in on this, if I just look at these 19 genes that are common between the two different types of wheat, we end up with a really lovely Netminer graph that highlights the cellular response to hypoxia as the common term between these. So this was an unguided network graph that independently derived to hypoxia stress. So this was great. So things like the glycerophosphodiester phosphodiesterases, there's inorganic pyrophosphatases, and a whole bunch of transcription factors. And these are things that we expected to know. Now, in addition to these, in the supplementary tables and figures, if you're interested, there's all of the lists of genes, and these are things that you can pull out and we can play with if you're interested in wheat's response to waterlogging. But coming back to blackgrass, there are many fewer differentially expressed genes in blackgrass, and they're much more varied. So although there was 23 in the Lola 103 and 27 in Peldon, there was only one that was commonly differentially expressed. So there are things like GSTs, there's PCB1, which is a really important waterlogging response gene. But the one gene that was commonly upregulated between these was OPR1. Now, OPR1 is a 12-oxophytodiionesic reductase 1, hence called OPR1, and it is involved in the first committed step towards jasmonate biosynthesis. The reason this is exciting is because jasmonates are involved in a huge number of metabolic responses, and it's a hormone that's involved in all sorts of biomolecular responses. But in addition to all of those other things, it was identified as one of the hub genes that's involved in non-target site resistance to herbicides. So this was a gene that was pulled out a while ago as being one of these really important genes involved in non-target site resistances. Now, because we knew quite a lot about the populations that I have been talking to you about in the field, these blackgrass lines, including 103, we had a good understanding of how they survived two different herbicides, phenoxaprop and cyproxidin. So when I plotted the correlations between waterlogging tolerance and phenoxaprop survival or cyproxidin survival, what you see is that there's not much of a correlation between cyproxidin. There's a clear correlation between phenoxaprop and waterlogging tolerance. Now, this is really great because phenoxaprop is survived by non-target site mechanisms, whereas the target site resistances are conferring resistance to cyproxidin. So this is great because it sort of builds out this idea that actually non-target site resistance and waterlogging tolerance are correlated with each other, that there's a suite of genes perhaps, or most specifically OPR1, that are leading to these resistances to both stresses. So perhaps just to remind you that non-target site resistance is conferred by a whole suite of different enzymes, all of which work together to take the herbicide and make it so that it is no longer able to cause harm in the plant. So as you go through the different phases, there's a reduction in the herbicidal activity. And if we look at the genes that have been associated with non-target site resistances, you can see that each of them is up in the waterlogged condition compared to the control condition. There's also a bunch of other genes that are up in waterlogging just as a comparative control. So this is really interesting because it's allowing us to say that perhaps there's a correlation between ability to survive herbicide resistance and ability to survive challenges such as overwhelming. Right, so bringing this all together, we've talked about the differences in the above ground physiology, that blackgrass is quite a lot bigger when it's waterlogged, whereas wheat is struggling when it's waterlogging. And perhaps this is due to differences in the root structure, where you have this closed root structure that has to respond and open up to make these parenchyma, whereas blackgrass has a root structure that's much more conducive to gas exchange in the beginning. And it's then therefore able to survive because it circumvents this hypoxia stress. This may explain the low number of metabolite changes that we see in blackgrass and the low number of gene expression changes that we're seeing. But it did identify that there's this nice correlation between ability to survive via non-target site resistance mechanisms and waterlogging mechanisms. So what does this all mean? Why is this important? Why are we working on this? So what are we learning from studying weeds in the agro-ecosystem? So on the food security side and how we are controlling weeds, what we've learned is that farmers need to take extra care to control blackgrass and wet patches of their field. This is quite important because we can feed this back and farmers can then use this information to make sure that they are targeting these wet patches rather than just spraying the field all over. On the other side of it, in terms of learning about successfully growing in suboptimal conditions, an open root structure and genes correlated with non-target site resistance allow blackgrass to avoid the hypoxia stress so that they can survive under these conditions. Now, this is all great. I've talked about this at a very high level, and I wanted just to bring it back because, as I said in the beginning, we're working with plants in a field, and we're not working in the lab most of the time. And we had to sort of work to bring agricultural weeds into the lab and develop them as model organisms. Weeds are great because they have lots of natural variation, they're experimentally retractable, they have super good fecundity, they have lots of relevance. But as we all know, for those of us who are logging into this call for any one reason or another, access to omics and ability to do reverse genetics is not as simple in weeds as it is in many other plant organisms. And I say this from firsthand experience because, as I said, I was trained as an Arabidopsis geneticist, and the work that I used to be able to do to easily get from a developmental output back to the molecular response in response to environmental input is much, much easier than the work that I'm doing on trying to understand how environmental challenges lead to survival by which molecular responses. So as we move from an agricultural setting like this, where we're wandering out into the fields and gathering seeds in the fields to try and get to a point at which we can have a DOI associated with a phenotype of interest, is not quite as straightforward as this. So I thought I'd just run through some of the things that we and others have done in order to be able to give this seminar sort of seamlessly, in that we did stuff and we came to answers. So first off, there's been a huge amount of effort at Rothamsted through the years to bring agricultural weeds into the fields, from the field setting into the labs. So for instance, colleagues of mine have gone out to farmers' fields, mapped where they are, how much they are, what's underpinning the soils and things, so that we can have these very well characterized germplasm from across England. This is absolutely essential for us to be able to do correlations between, for instance, waterlogging tolerance and phenoxypop survival. They also allow us to do things like ask whether or not there are correlations between herbicide resistance and other life history traits, such as flowering height or head number or time to flowering. And the nice thing I just wanted to highlight here is these analyses have allowed us to show that there are no correlations between herbicide resistance traits and life history traits, even though we're seeing clear tolerances between waterlogging and survival. So some things are going to be linked and some things aren't. Now, I also said that we are bringing weeds in and studying them in the glasshouse. We needed to generate the protocols in order to analyze these weeds root sections and to try and see what was going on here. And a lot of this was a lot of trial and error. I have to say a huge thanks to Christian Harrison, who spent a lot of time trying to kill blackgrass. He's got a green thumb and he was not able to kill wheat or blackgrass, but he finally settled on this wonderful setting where we were able to induce waterlogging in a controlled way. Now, I've also just sort of said, oh yeah, we did some transcriptomics. And of course, in order to do transcriptomics, we need to have a genome in which to do that. And so this was a huge effort in order to get a platinum quality genome that allowed us to do things like map our transcriptomics back onto them so that we could identify exactly which gene was being upregulated when in response to these treatments. So a team from Clemson, Bayer, Rothamsted, and the University of Copenhagen put this together. This came out in the end of 2023. And the genome not only allows us to do things like say that there are 15% of DNA transposons in the genome, but also it allowed us to say that there's evidence for both parallel and non-parallel evolution of herbicide resistance. So working with the lines that were brought in from the field, we did some controlled crosses, and they allowed us to map QTL regions and differentially expressed genes for 2 different families. This was really cool because it allows us to show that the QTLs do not overlap. So these purple lines do not overlap with any of the green lines. This is just a snapshot of one of the figures from the paper. And so this showed that there is non-parallel evidence for generation of herbicide resistance. However, of the 643 differentially expressed genes, about 10% of them were overlapping. So there are some outputs from those different herbicide resistances that are in parallel. So it's not all that they're just totally different things, but there are some underpinning things that are consistent across them. Another thing that I don't have much time to get into, but we have evidence also for extra chromosomal circular DNA in blackgrass. So we use these same mapping populations in the same controlled process to look for evidence of ECC DNAs in blackgrass. We found them, it was very exciting, and many of them have cDNAs on them. This is quite unique amongst species that the plants have them. You might have heard of ECC DNAs as they're involved in herbicide-resistant polymer. These are extra chromosomal circular DNAs that float around in the cytoplasm, and they're involved in herbicide resistance in palmer as compared to cotton, and they're also inherited in lovely little ways. Coming back to blackgrass, we had some extensive analysis of p-family IDs showing what is shared, what is unique, what's overlapping. Again, what we're seeing here is that there's some parallel and non-parallel evidence of herbicide resistances being targeted within these populations. And one of the things that keeps falling out is that GSTs, glutathione S transferases, and of course OPR1 has come up in multiple different times across these analyses. OPR1 is also on our ECC DNAs. So this is a gene that keeps coming up over and over again, as is GSTF1. And I've not too much talked about GSTF1, but I'll spend a little bit of time on it now. But as we all know, ice creams and shark attacks are correlated, but we can all agree that they're probably not causative. And so just getting to this idea that testing whether or not these genes are coming up over and over again, are they actually causing non-target site resistance, or are they just correlated? So GSTF1 has come up in a huge number of populations, a huge number of publications, and we have a good understanding of what it is. It's also come up in our analyses. So GSTF1 is this highly expressed gene here that has come up again and again. And so what it does is it conjugates glutathione onto your target, and so you end up with a thing that no longer works. Just like if you stuck wheels underneath a doorstop, the doorstop wouldn't be as functional. That's sort of an equivalent for what GSTs are doing. So I asked, what happens if we could bring that down? What would happen here? Now, the question is, will this go from a plant that is herbicide resistant to one that is sensitive again? Now, if I was doing this in Arabidopsis, it would be super easy. We're not doing it in Arabidopsis, so I had to do it the hard way. And the hard way was actually to take a virus-induced gene silencing and to drive the expression of the GSTF1 by this virus-induced gene silencing. And what you can clearly see is you have a resistant plant that is now more sensitive. We can also use this the other way around and take a sensitive plant and make it resistant by driving a different gene by virus-induced overexpression. So this is VOX bar. And so these virus-mediated reverse genetics allow us to ask questions about what's happening here. So if you are interested in the mechanisms of this, there's a paper that I put out in Pest Management a few years ago now, and it goes through all the details. But essentially, in essence, what we're using is we're using the virus to trick the plant to either make genes of interest for VOX, so it's tricking the plant into making a new gene that comes out for a new protein, or we're tricking the plant to knock down its own endogenous gene by making it think that the gene is coming from the virus. So this not only allows us to do cool things with the herbicide resistances, it also allows us to do cool things like make the plants grow. And so this is VOX driving GFP and blackgrass, or we can make stripy plants where we've knocked out phytoene desaturase and you end up with a plant that's no longer able to produce chlorophyll. So it is tricks and tips, but tricks and tips are often very useful for asking hypothesis-led questions about what does a gene do in the gene functions. So essentially, in summary, this is allowing us to test hypotheses about gene function in the plant of interest, in a whole plant of interest. So you can ask whether or not dead plants can be brought back to life or vice versa by changing the gene expression in this plant. Now, as Kaio mentioned, I'm not only interested in Alpharis myricetes and blackgrass, I also have other interests in terms of orange foxtail, various lowlands, some palmers, some amaranthus, and chenopodiums. And the reason that I'm doing this is because although I said that blackgrass is right across Western Europe here, there's a lot more world and there's a lot more weeds across the world. So what I'm essentially doing is I've challenged the lab to derive a mechanistic understanding of weediness. And I mean, again, by weediness, weeds come in all different shapes and sizes, all different flavors, but essentially weeds are things that humans don't like. So if we can try to figure out why they're surviving, then we can try and figure out ways to study them. So we have a variety of different techniques that we're using. Some are more molecular than others. We're also then bringing this back around, centering it on the field, because again, we're bringing this back to the agricultural relevance for why we're studying these so that we can have more food and essentially just more cake. So working this back, if we want to ensure food security, we must understand better weeds. If you know the enemy and you know yourself, then you need not fear the results of 100 battles. Hopefully over the course of today, I've convinced you that weeds are our enemy and that there's a clear correlation between more weeds and less food. I've introduced myself as a molecular geneticist, and I'm working across this landscape to genes sort of circle and trying to approach to derive a mechanistic understanding of weediness and how they're surviving. And I think regardless of where you're tuning in from today, there will be a color in your country of interest. And this is probably correlating because everybody has a problem with weeds. And all of us need to try and better understand our weeds so that we can ensure food security. I already showed this slide once, but again, this is a huge collaborative effort. I want to thank everybody that's been involved in all of these processes across all of these. However, the people that are most important to today's talks are Christian, Clarice, Gianluca, Jane, David, Dan, Andrew, and Sigrid, all of whom are on this paper, which came out in Functional Plant Biology earlier this year. And here's the link for it. I'd like to also acknowledge Growing Health and Rothamsted Research, all of the teams with whom I'm working, and special shout out to my 3 PhD students, Peggy, Gerda, and Tala. And hopefully they will be the ones who are delivering the next seminar rather than me, as they have lots of exciting, lovely research that they're starting to come out with. That's the end of my slides. So what I will do is I will stop sharing and I will hope that there's still some, there you are, excellent. So hopefully that's now stopped sharing and I'd be happy to answer some questions if there are any.
Thank you, Dana, so much for fascinating work sharing with us. Appreciate it. I will break the ice. I will ask you the first question. I've been thinking over your presentation here about this correlations between abiotic stress and abiotic stress tolerance with herbicides. And do you think that that might be something we are, as weed scientists, overlooking, and that it's more important than we previously acknowledged? For instance, over the thousands of years that plants have been exposed to abiotic stress, heat, drought, things like that, maybe that has a greater association with herbicide resistance nowadays that we are just seeing now because over the last 50 years we're spraying herbicides.
I think scientists in general like one-to-one correlations. You know, we like cause and effect, we like application and death or not death, as the case may be. And I think as scientists, we tend to break things down very, very systematically so that we only look at one process. I think that it's unrealistic to think that herbicide resistances are just coming up in the last 50 years. I mean, plants have been dealing with challenges since the dawn of time, and since agriculture, they've been having to survive under these situations. I suspect the more we look, that there's going to be more correlations between herbicide tolerances and all sorts of stress responses. We already know that some of the genes that we're looking at are naturally evolved to help plants to survive generally, and that weeds are just better at taking these on. So for instance, in the genome of blackgrass, we have a huge expansion of GSTs compared to other plants. So of course, we're going to find GSTs are involved in herbicide resistances because these are stress tolerance mechanisms that are coming up. I hope I haven't showed all of my secrets in terms of the correlations between stress resistances and abiotic stress and the other tolerances. But I do think that scientists in general need to take a step back and say, what else is happening in my plant, rather than just in my lab, when I do this, that happens.
Yeah, cool, thanks. And we have a few questions here in the chat, so I'm gonna read them. Is it possible to expect at least a minimal trade-off between hypoxia tolerance and blackgrass, given that the tolerance is inherent, it seems that there is a significant rearrangement of root structure, which should need extra energy from somewhere else?
Yeah, so one of the things that's really cool about the differences in the root structure, there are drawbacks to having that open root structure. Of course, that's why some plants have it and some plants don't. However, the cool thing about blackgrass having this open root structure is that it can kind of ignore that hypoxia stress, because it's able to do the gas exchange without reconfiguring its entire root structure. It can take that metabolic energy and put it into growth. It can take that extra water, take that extra energy that it's getting from the nutrients it's picking up, and grow bigger. Whereas wheat has to kind of stop, realize it's being structured, do some programmed cell death, and restructure its root structure. Without, it has to then pause its growth in order to use that metabolic energy elsewhere. Now, there might be some drawbacks to having that open root structure as compared to the closed root structure, but that's something that we can look into more as to exactly if there are any drawbacks or if there are any detrimental traits.
Oh, thanks. Another question, what type of non-target site resistance was waterlogging resistance in blackgrass associated with?
Yeah, so it's really specifically the non-target sort of metabolic resistances. So there's as the question, thank you, I skimmed over quite a lot of what was happening in non-target site resistance. It's basically everything that isn't target site resistance. And that catch-all means that there's things like changes in absorption, translocation, all sorts of things. Really, this is the metabolic aspects of non-target site resistance. So this is specifically changes to adapting the herbicide molecule so that it has different processes so that it's not able to so it's either more sequestered or so that it's less toxic to the plant. So OPR1, there's a clear correlation. The GSTs did come up a lot. I didn't get any cytochrome P450s, but that's probably just because we had very few differentially expressed genes, more than we had, that it's not happening. I think if I had an earlier time point where I was only looking at that sort of immediate shock response, I suspect the cytochromes would come up. But yeah, really OPR1 was the main target. Some of the GSTs did come up as well.
That was a question I had. How did you get so few differentially expressed genes? Did you have any control crosses or something like that?
Yeah, so the differentially expressed genes were compared to the control. Because the hypoxia stress is partly being avoided because of the physiological differences, blackgrass is kind of just doing its thing and not really responding to the waterlogging stress. So because the hypoxia stress isn't being induced to the same degree as it is in wheat, then the responses are much more muted. It may also just be that by the time we've done the measurements, that most of that big peak of responses has then dimmed out. We're not quite sure. I'd have to go back and do an earlier time point in order to analyze that.
Yeah, that makes sense. Another question is, are you able to transform blackgrass? There are protocols out there, and you talked a little bit about it.
Many years of my life trying to figure out how to transform blackgrass. I'm still working on it. I haven't let that one die yet. So instead of doing a stable transformation where I'm taking a gene and putting it to the genome so that it's able to be persisting through the time, I've decided to use these virus-induced gene silencing and virus-mediated overexpression, so VIGs and VOX. The beauty of this is that it happens within the generation of the plant. It's transient, so that it's easier to control and easier to moderate. But also it's quick, it's fast, it's easy. It answers the question in a way that doesn't take me 3 years of doing callus transformation, making sure that I regenerate the plants, then crossing them backwards and making sure that, so the process of a stable transformation in blackgrass is still in process, hopefully, but there is no process right now. But the VIGS is great because it allows us to change gene expression in the plant of interest in less than 50 days. So you can go from construct to phenotype, an herbicide phenotype in 50 days. So that's really powerful. It's really strong. It's working at high efficiency.
Yeah, that's awesome. That's not much, not very long, 60 days, to be
No,
honest.
it's quick.
You don't have to do any controlled crosses, anything like that. Another question, should wheat breeders try to bring some blackgrass genetics into wheat breeding into wheat genome? Are they doing some of this?
I had really hoped that there would be some fantastic gene that would fall out of the screen and we could say, put this pathway into wheat and you will generate waterlogging tolerant wheat. OPR1 is already highly expressed in wheat. We already know about jasmonate biosynthesis and its links to stress tolerance. Had there been something super novel, super interesting that had come out of the blackgrass genome, absolutely, I would be on the phone to the wheat breeders and I would say, this is what you need to tolerate. But these are the sorts of possibilities that we can open up by finding the genetics of our weed species and studying their tolerances. If there had been something, yes, of course. Unfortunately, what came out was OPR1, so it's slightly disappointing, but at least it proves the point that we can identify pathways and we can identify genes that the breeders in the future could select for if we could find something that was cool and interesting and different.
Yeah, awesome. And the last one is more like a comment talking about great presentation, thanking you for sharing with us. And they think that the focus on weed biology, understanding differences between weeds should be to better ensure that we don't have to be more reactive, instead understand the weeds from the beginning so we can better plan our control strategies.
I'm very, as I said, I'm very much on the plant side. And so I often apologize to farmers for being very excited that they have huge blackgrass pressures in their field. I think that weeds are resilient. They're really able to survive and thrive in our agri-ecosystem. If we could take some of that and move it into our crops, we would be less worried about climate change. We would be less worried about food security. We would be less concerned that there's a new something that's going to come on and wipe out our food security. I think if we can better understand how plants, intentionally saying plants, not weeds, but how plants thrive, then we can use that information going forward.
There's one last question here asking, do you think wheat with constitutive open arrhythmia like blackgrass will have negative effect on traits such as yield? Do we have a mutant for wheat with arrhythmia?
Yeah, so in rice and in wheat, that formation of arrhythmia is a responsive trait. So we've seen that in a number of different species where in response to waterlogging, they create these arrhythmia. They don't have them constitutively. So there may be some trade-off between perhaps, now I haven't looked at how these guys do under other conditions. So there might be other conditions under which I can challenge them and they would not be as successful. We can systematically start to work through that. But there's some interesting things that we could start to analyze, but in other species, they are not there unless they're needed, which implies that there is some trade-off, at least in the crop species that we work with. There's some trade-off that it's better to have a closed root structure than it is an open root structure. If you think about what roots are doing, they're moving things back and forth. They're important for structure. They're important for density. So having this giant hole in the middle of your root probably is not great. It works for blackgrass. It works for other species in response to waterlogging. I don't know. I'd be very happy to try and hear some suggestions about what other stresses you would like me to ask. I'm not so interested in looking at things like yield in blackgrass because that's not really what we're looking for. But really, that growth and that ability to survive is more, I think, an immediate response rather than just in the yield. But yeah, I'm very happy to talk to people if they want to challenge me with how else I should try and kill my blackgrass.
Well, thank you so much, Dan. I appreciate your time. Do you have any final thoughts to share with the audience?
No, I thank you all for coming. I really appreciate this. I think that the seminar series has been really helpful in trying to engage with people about why we're doing genomics and genetics. And I think that hopefully what I've showed you guys today is that having genomes and having the access to doing hypothesis-led research in the lab really can allow you to explore questions that you wouldn't otherwise think to explore. So why is this not dying rather than just, is it dead or not dead when under these conditions?
Yeah, awesome. Thanks so much. It's good to see you. Thanks so much. Have a good rest of your day.
Thanks very much.
Thank you. Thanks everybody for joining us today. And we were going to be sharing more information on the upcoming webinars. Thanks so much.